Hello, I am Jitendrabhai Zaveri, and I am currently a sophomore at West Virginia University. I study computer science and neuroscience. First of all, I want to thank my mentor, my advisor, Dr. Gianfranco Doretto, and the other main contributors, Keaton, Meghana Kovur, and Henderson, for allowing me to present our research here. Our research is plant image analysis in the wild. There are a lot of applications available in the market to classify plant images, but they have a lot of requirements, and for that we want to make our algorithm more generalizable to the real world conditions, and that is the objective of our research. So let's begin with some introduction. This is the computer vision problem, and for that we are using machine learning. These are just applications of artificial intelligence. In computer vision, computers learn and understand, extract information from images and from videos. And machine learning, in which computers are to learn automatically without human assistance. If you can think about real world applications, there are autonomous vehicles available, and there are also face recognition algorithms in our phones. On this project, we're utilizing two of the machine learning approaches, which are classification and object detection. So in classification, we want to classify an image according to its respected class. For example, we have an image of a cat, so we want to classify that image as a cat. And in object detection, we want to localize and detect objects in an image. So there might be multiple objects, so we want to detect each one of them and locate them, and also classify those objects. So this example shows how classification and object detection works. So here we are going one more step deeper and utilizing fine-grained visual classification. So this is used when the categories are very similar, visually very similar. So for example, if face recognition, the faces are very similar, so it's kind of hard to classify those using normal algorithms, normal models. So that's when we use fine-grained visual classification. Um, as you can see in the example, there are leaves. They have similar color, shapes. So they are visually very similar, and that's the reason we want to use fine-grained visual classification technique here. Now let's talk about the challenge and the data set we are using. This is a challenge from PlantClave, our initiative from ImageClave Research. They provide dataset for each competition year. So the dataset we are using for this research is from the year 2015. This data is collected by untrained individuals, just like citizen science projects. So they often contain a lot of background noise. They also vary in color, some might be blurry. They also contain major occlusions, etc. So that's the reason the task is challenging. The dataset contains over 100,000 images, which are focusing on 1,000 herb trees and fern species. Each image has multiple organs in it, so we are annotating bounding boxes for each of these organs, which include leaves, flowers, fruits, stems, and the regions where the leaves are in high density, which we call HDL. After all of the annotations are done, we derive the annotations count per organ. So as you can see in the graph, leaf has the highest number of annotations than flower, fruit, and so on. We talked about annotations in previous slides. For that task, we used a tool called Snappy Annotator, which was developed in our lab. So that's how it worked. We label them in the organs, and we export those to an XML file and use those to derive the information from it. Our approach is very simple and straightforward. So first of all, we are processing an image. It will detect the organs, each individual organ from that image, and they will process that into the respected species classifier. So in this diagram we can see that it detected leaves, and then it processing those to leaf classifier, and then once it's processed all of the classifier will have the confidence score generated. So we'll fuse those confidence score and then develop that information fusion, and then we will have the final predictions according to a particular threshold. For object detection, in our case, organ detection, we are using faster RCNN model. It's actually based on Detectron 2 code developed by Facebook Research. Before I go to the next slide, I want to mention that we are using ResNet models. Currently, they're very popular and very powerful classification models. So as you can see, there are various models in the series. The number shows the depth of the model. For organ species classification, we are using ResNet 18 model. There will be a species classifier for each organ. Currently, we are only processing those classes with the highest amount of annotations, and we might need to increase the number of layers. So we might need to use ResNet 34 or deeper models if we increase the number of classes. As I mentioned earlier, we'll have confidence scores generated from each classifier, and we'll fuse it together to predict a final prediction. And for that, we are using information fusion technique. So this is a total overview of our model. We have tested our model and received the preliminary results for object detection and species classifier. For this preliminary results for object detection, we trained on the images containing only leaves, and we processed the images and got the results. As you can see in the image, on the right side it's ground truth, on the left side is predicted image. So it almost predicted precisely for this example. And here we can see that the ground truth contains two leaves, but in the predicted image it predicted many different areas and many different leaves, and which are not leaves. So our model has a lot of flaws that we might need to perform a lot of optimization on, considering we had the maximum amount of annotations in our plant organ leaf. We chose that to show our preliminary result. So we trained our classification model on the first 40 species, which contained the maximum amount of leaf images, and we received this result. As you can see in the confusion matrix, there is a clear diagonal. So some of them contained over 500 images, some of them contained around 100. So we still have a little flaw in this matrix, but the accuracy was pretty high for these particular tasks, and we are inclined to use some deeper models for more species classification. We are yet to implement our full model on the images and the organs, and for that reason we have not developed information fusion yet. I especially want to thank Stanislav Podolsky for assisting us with the development of annotation tool Snappy Annotator. Furthermore, this material is based upon work supported part by National Science Foundation. So if you have any sort of questions about models or about results or approach, please put that in the comment box, and I'll be more than happy to answer those in real time. Thank you.